Shall be getting the PDF soon, despite the Canvas nightmare issue. So I'm going to start this, and as everyone is aware, we do have another exam coming up, right? Very quickly. That's going to be exam number 3, and I believe it is on Monday, right? Okay, just checking if you know when it is. Okay. And the most asked question is, what is on the test? So it's going to be from where we left off last time, covering all the way to muscle, to the subject that I'm going to cover also today and on Wednesday. Okay, so that's all fair game for the exam next Monday. All right. Also, I've seen your emails, asking for opening of the Pearson for Chapter 8. Yes, I will open. I usually open again those assignments until the exam date that includes the assignments. So, I'm going to be opening those assignments again as soon as we have access. Yeah, okay. So getting here into the muscle. What we're going to look at, of course, at the gross anatomy. Then we're going to look at microscopic anatomy, and we are going to look at what happens, what is the process that has to go on for your muscles to contract, right? So we are going to look at what is sending the nerve impulse from your brain down to the neuromuscular junction, and then chemicals that are important for muscle contraction and all the good stuff. So it's a lot of information in these 2 classes, right, today and Wednesday. So I will go into as many details as possible, keeping in mind that I like to tell a story, not just dump the information over your heads, right? So hopefully it's going to be clear and understandable. Okay. And if we do have time Wednesday, I'll tell you a very cool story about absolute refractory period. Okay. Unless you have already watched it in my videos. But anyway, I'll tell you about it. So here, when you were talking about skeletal muscle, as we started talk briefly before, we are talking. And now I forgot to close my email. Okay, hold your thoughts. All right, so continuing the thought. As we have talked about before briefly, when you want to move something, you're going to depend on a muscle, and the part that you are moving is the insertion point, right? So if you're using your biceps to move your forearm, the bones here on your forearm are your insertion points, and the bone that doesn't really move much, that is your origin. Okay, so origin not moving much, insertion point, insertion is the part that moves. Okay. So how is it that you are going to connect muscles to mainly bones, right, in order for something to move? You have 2 ways of connecting the muscles to the bones. One way is a direct connection, which is not as common as the other way, which is the indirect connection. Okay. So when you're talking about direct connection, what you're going to see here is the connection of the outer fiber that covers up the muscle directly to the fiber that covers up the bone. So in a direct connection, you are going to have the epimysium from the muscle that is going to really get together and fuse to the periosteum of the bone, in case it is connecting to a bone. In case it's not connecting to a bone, in case it's connecting to cartilage, you're going to have direct fusion of the epimysium of the muscle to the perichondrium of the cartilage. So remember, peri is the membrane that goes outside, right? And osteum has to do with bone, and chondrium has to do with cartilage. So this is the form of direct connection from the outer membrane of what is encasing the muscle to the outer membrane of what is encasing either the cartilage or the bone. Now, the most common form of connection that we have is actually indirect connection. So now you're going to have the outside part of your muscle that is going to be covered by this other type of fiber that is going to extend and originate tendons. Okay, so the tendons are then going to connect with the bone. Now, similarly, you have, instead of just one direction tendon, you can have a whole sheath that can connect to bones or to other organs, and the sheaths are called aponeurosis. Okay, so if it is a rope-like structure, that is a tendon. If it is a sorry, Portuguese problems, like sheet of paper, that is the aponeurosis thing. I can never say the right sheet. Okay, so here. All right. So these are the connections. So 1 and 2. 2 is the most common one. All right. Then we're going to start looking at the skeletal fibers. Okay. So fibers is the same, is the name that is given to a muscle cell. So if you say, is it a muscle cell or is it a muscle fiber? Well, it's basically the same thing. Now, when you look at a muscle fiber, you'll see that it has certain properties, right? It has to respond to some kind of a stimulus. It has to have a quick response, okay? And it has to be a significant response. So we say these muscle fibers, they have to be fast, they have to respond, right? And they have to have some powerful way of responding, otherwise you don't really see movement, right? And another characteristic of skeletal muscle fibers is that they can get tired, okay? They can get fatigued or what do you call it? They can get into a state of hyperstimulation or tetanus state. And sometimes, they can kind of like give out for a little time in case the muscles are not quite tuned. So they have to respond, and they get to be a powerful response, and they're going to get tired, the skeletal muscles. And another very important characteristic is that they are all voluntary muscles. So at your will, you're going to contract a skeletal muscle. Okay. All right. So here, macro structure of your skeletal muscle. Here you see a bone. Okay. Over. Okay. Okay. Here, it's a bone, right? So here we see that you have this type of fiber that connects to the outside of this muscle stuff here. Okay, so this is on the outside of the muscle, and this goes like basically on a one direction connection, like a rope to the bone. Okay, now you can imagine the type of this fiber has to be dense, right? And since this is a rope-like structure, this is mainly regular, dense connective tissue. Okay, because it's going to pull it like a rope in one direction. So you want this thing to be quite strong. All right. So after this coming together with this tendon, extension of this structure, you're going to find this epimysium structure. Okay, so this epimysium here is going to be on the outside of your muscle. Okay, and this is going to be made of dense but irregular type of connective tissue. Okay, so besides that, so this is going to go all around the muscle. Now, continuous with your epimysium is going to be this other fiber here that is called the perimysium. Okay, so this here is going to go around these bundles. Okay, around these bundles of muscle cells. Okay, so epimysium is also made of dense, irregular connective tissue. Is a tough type of fiber. Okay. So these bundles here are going to have a lot of muscle fibers or a lot of muscle cells that are bundled together. Now, around each of the muscle cells, you are going to have another layer that is called your endomysium. Okay? So endomysium is not the membrane of the cell itself. It's another layer that goes around the cell membrane. Okay? So this endomysium is of a different type of connective tissue that you find in the perimysium and in the epimysium. So this endomysium is more of a not dense, more of irregular type of tissue that, of course, I should forget the exact word, but it's going to come out soon. But is more fragile in comparison to the perimysium and the epimysium. One important thing to keep in mind is that all of these connective tissues, they are actually connected. Okay, so it is a continuum of connection between this outer layer and the middle layer and the inner layer of those connective tissues. Because if you think about what is really the function of the muscle, of course, is to contract, is to move things around. But what is going to contract is going to be little fibers that are inside of this muscle cell. So when the fibers contract, if you don't have a continuum attachment of these fibers to the outer layer here, to the endomysium, and then to the perimysium, and then transferring that force to the epimysium, if you don't have a continuous transfer of that force, then nothing is going to happen. You can try to contract your muscle as much as you want, you can contract your muscle fibers, but if you don't have transmittance of that force, nothing is going to move. Now, with that in mind, you can think that if you have problems of attachments between muscle fibers and the epimysium, you might have problems with muscle contractions. Can you think of a disease, or does anything come to mind that has to do with problems contracting muscles? What kind of disease would you think? I don't have a disease in the bag. I actually have incentive, chocolate. But yeah. Huh? That is more of a problem of the nerve. Multiple sclerosis is more of a problem of peripheral nerves. But you'll get a chocolate. Right? Oh, sorry. They don't fly as well as the nuggets. I'll get them on Wednesday. Yes. Myopathy is a problem disease of the muscle itself, but it doesn't really have to do with the connection with these layers. But you got a chocolate. Oh, I got it. Okay. It sounds like muscular. Yes. Okay. Ready? Muscular dystrophy. Yes. And you got one too here. No? Oh, just in case. Here we go. All right. So, muscular dystrophy. As we're going to see, there is a name of a protein that's called dystrophin. Then when the protein doesn't form properly, you lose the connection that should be transferring that force from the inner fibers to the outer fibers, right? And that protein is important for this overall transfer of energy. So if you don't have that thing working properly, you end up with muscular dystrophy, right? So having this idea in mind that when you contract the tiny thing here, this force has to be transmitted to the outside, right? So now we're going to see more about that, right? So here is a slide showing you this aponeurosis. So this is a muscle, okay, this thing is muscle. So here is the connection of connective tissue sheets, okay, this aponeurosis. Okay, mainly it's showing you it's similar to tendons, but it's like a sheet-like 2D instead of 1D. All right. Other types of muscles that you have, as we mentioned before, you have your cardiac muscles. And where is it that you're going to find a cardiac muscle? Is only one place, is only in your heart. Okay. They are also striated, like you find with your skeletal muscles. The big difference is that this muscle is involuntary, so you cannot at will speed up or slow down your heart. Okay. Another difference is that you have branches that are connecting those muscle fibers. It's not just like a parallel position as you have with the skeletal muscles. Okay, but those are also of course striated. Okay. The third type of muscle that you have is your smooth muscle. Those are also involuntary. And the difference between this and your cardiac muscle is that these guys are not striated. Okay, where you find those, mainly you're going to find them in walls of hollow organs. Okay, so here is showing your stomach. You find it also in your esophagus and intestines, and you also find in the walls of your arteries, mainly your arteries, so the vessels that are coming out away from your heart. There is also a bit of smooth muscle in your veins, but not to the degree that you find in arteries. Right, so this is a chart from your book showing the comparisons of these three muscle types. So we have seen that before. And here you come for your first question. And I'm putting here the level of confidence on. And this is just to warm up quite kind of a simple question, right? Of the following muscle types, which is the only one subject to conscious control? Yeah, that is like too easy of a question. Let's just make sure that our eye clickers are working. Everybody's able to connect and get your extra credit points. Okay, does anybody need more time? No? Everybody got your points? Okay, I'm gonna stop it. All right, what is your answer? B. Yeah, and I did have people who told me cardiac. If you think it's the cardiac muscle, talk to me after class. All right. So here, 4 important muscle functions. One, of course, is movement. Okay. Another important muscle function is maintenance of posture. So although you are not really thinking about contracting your back muscles right now, you are contracting them, so you are actually sitting, right? If you don't contract those back muscles, you're going to just flop down and fall off the chair. So maintenance of posture is an important function. And interestingly, those posture maintenance back muscles, they don't really get fatigued. So it's a good thing because you got to be sitting up or standing up or doing things that are actually out of bed most of the day. So if your back muscles will get fatigued, that will be a problem. Right? So maintenance of posture. Also, important function here is to stabilize joints. So besides all those things that are involved in joint stabilization, like tendons, like ligaments, right? Ligaments connecting bone to bone. Muscles are very, very important for joint stabilization. And another factor that we really don't worry about it much in Florida is generation of heat. right? So when it gets really, really cold and you go outside and it's like shivering. So a lot of people are going to travel up north for the holidays, and it's going to start getting cold up there, right? So you step outside and you're used to Florida weather, and you go like, oh, it's 60 degrees, it's freezing. No, it's not freezing, but you're going to start shivering, right? Because your muscles want to adapt your body temperature to what you're used to, like in Florida, right? So generation of heat is quite important function. All right. So here, characteristics of muscle tissue. Okay. First characteristic is that they have to respond to certain stimulus. We call that excitability. Okay, so responsiveness. Another thing they have to do, they actually have to contract. That's why you call them muscle tissues, right? They have to contract. This third characteristic is interesting because it has to do with extending the muscle, ability of this muscle to stretch. So you can stretch the muscle after. If you have it in resting position, you're actually able to stretch it a little bit, like not too much, but you're able to stretch it a little bit, okay? And the fourth characteristic here is that it tends to go back to its resting position if you're not contracting or extending it. So that is the elasticity of the tissue. So what you find in the muscle? Well, you're going to find a very good supply of blood, and also you're going to have a lot of nervous endings, so a lot of nerve supply. Because if those muscles that we're talking about, the skeletal muscles, if you're going to command them to contract via a neuromuscular junction, you actually have, of course. So you actually have to have the nerve that is going to get to your muscle. Okay, so a lot of nerves to supply the impulse from your brain to your muscle. So of course you need to have the nerve, and why do you need a lot of blood in the supply? Yeah, you need to provide oxygen, and you need to provide nutrients that are going to allow this muscle to have enough juice to contract. Ooh, I found a nugget, right? Oh, that flies so much better. I gotta bring those to class next time. All right, so here. All right, so here is just a write-out of what you need to know about those sheets of connective tissues, right, that are enveloping the muscle. Remember the order. So you have that epimysium that's most on the outside, then perimysium in the middle, endomysium surrounding each nerve fiber. Okay. All right, so here's the word. These two here are dense connective tissue, and this endomysium is actually that areolar, fine areolar type of connective tissue. All right, so we have seen that. Let me see if I missed something. No. Okay. All right, and I like this picture here. They actually don't remember where I got it from. That is showing the different layers. So here, okay, you have this bundle of sheets of fibers that are together with the whole muscle, right? So when you look at down to the individual cell, okay, so when you look down to the individual cell, you're going to find your endomysium, like right here, surrounding. So this line here is also going to down here, surrounding the fiber, okay? And under that endomysium, you're going to find your cell membrane. So your cell membrane is called sarcolemma. Okay, so muscle cell membrane is called sarcolemma. Okay. Now, inside the cell membrane, you're going to have your cytoplasm, right? But for the muscle cells, you don't just call the cytoplasm. You call that a sarcoplasm. Okay, so sarcolemma for the cell membrane, and sarcoplasm, of course, for your cell cytoplasm. Now, inside of that cytoplasm, this is where you are going to find those little fibers, those myofibrils. Okay, and then the myofibrils are going to be arranged in a certain way that, as we're going to see, they can slide on each other, and they can generate the contraction, the muscle contraction. Okay, so I didn't really find a picture in our book that was showing this arrangement, because sometimes it's quite easy to mess up, or to think that the endomysium is really the sarcolemma when one is not. Okay, so sarcolemma is the cell membrane. And remember, your skeletal muscle has a lot of nuclei. It's not only one nucleus. Okay, when compared to cardiac muscle. So skeletal muscle is going to have a lot of nuclei along this very, very long cell or fiber. Okay. Oh, I had it animated. Down to the myofiber. All right, so this is the picture from your book. Okay. All right, so here is what I just mentioned about your sarcolemma, for membrane, okay? And then your cytoplasm, of course. And inside the sarcoplasm, you are going to find other things that are also very characteristic of muscle fibers. Like, for example, you're going to have a lot of storage of glycogen, and you call those storage vesicles, you call them glycosomes. Okay, so they're going to store glycogen. You know what's glycogen? Is kind of a tree of sugar, of glucose. Okay, is almost like starch, but the branching is different between starch and glycogen. Okay, so this is just storage of glucose inside of the muscle. Okay, so besides those two, you're going to have an important protein that is called myoglobin. Right? So myoglobin is like a hemoglobin, except that myoglobin has only one polypeptide chain instead of 4 polypeptides as you find in hemoglobin. Now, myoglobin is really, really good at binding to oxygen when oxygen is delivered to the muscle via the blood, via hemoglobin. All right, so when hemoglobin gets there with oxygen, myoglobin grabs the oxygen quite quickly away from the hemoglobin. Right? So you're going to have store for energy in glycosomes, and you're going to have also myoglobin that is able to grab oxygen very easily. Okay. So here are names also of modified organelles that you're going to find in fibers. Okay, you're going to have those myofibrils, and those are the ones that are going to allow for muscle contraction and their modification of the cytoskeleton. Okay, so modified, specialized versions of cytoskeleton type of fibers. Then you're going to have here the sarcoplasmic reticulum. And this is a modification of your smooth endoplasmic reticulum. So your sarcoplasmic reticulum is going to be very, very important for storage of calcium, because you're going to need calcium if you're going to contract those muscles. All right? So besides that, you're also going to see these things that are called T-tubules. And these T-tubules are going to penetrate between cells, between fibers, and they're important for transmission of your nerve impulse, okay, in order to allow for excitability of your cell and contraction. But we're going to make it more clear, not just dump all this information at once, okay? All right, so here, let's look here at this whole thing here is your muscle cell, which is very, very long, all right? Now, each one of those muscle cells can have a lot of these myofibrils, okay? Like how many? Like, well, thousands of these myofibrils. And they're gonna fill up this cytoplasm. They're gonna take up most of the cytoplasm, like 80% of that cytoplasm is going to be made up of these myofibrils. Okay. All right. So when you look deeper in the myofibril, you're going to see those lines that we call them striations, okay? And here is where you're going to see something that looks like a fat line, a darker line. So you call that the age zone, okay? So this is going to be the fat dark line. And you're going to find skinny lines that you're going to call eye zones or light zones. We have better pictures for that. All right, so here we go. So that will bring us to the sarcomere. So sarcomere is really the smallest functional unit of your muscle cell, okay? And what is a sarcomere? Well, it's basically defined as this space that has a dark zone, okay? And it has light zones. And in the middle of the light zones, you're going to have this Z disc that holds your skinny fibers together, okay? So this Z disc that is holding your skinny fibers, from one side to the other side, that you find another Z disc, and it's called the Z disc because the arrangement of these proteins look like a zigzag line, okay? So between these two bands, you have your sarcomere. So sarcomere defined as between two Z discs, okay? So you have your eye, there is a light band. In the middle of your eye band, you have your Z disc, and then from one to another, that is your sarcomere. All right. Now we look at the myofilaments. So those guys are interesting, right? So you're going to have fat filaments and you're going to have skinny filaments. The fat filaments, those are going to be your myosin filaments. The skinny ones are going to be actin filaments, right? But there is more to the story. Okay. So when you look here at your myosin filaments, you're going to see that they are joined together by their tails, okay? And some of these myosin heads are pointing to one side, and some of those myosin heads are pointing to the other side. They are joined together at their tails, and the part that joins them is this M line, joining the tails of your myosin, okay? Myosin fibers here. Now, at the other side, you have some elastic proteins that are going to connect your myosin to your Z disc proteins, okay? So you see here this little coiled thing that tells you this is an elastic type of protein, okay? And you call this elastic protein a titin, okay? So this guy is titin. He's going to keep this thing held together when you have muscle contraction, okay? Right, so let's see here. All right, so looking more here into your thick filaments, those are myosin filaments. Each myosin fiber, each myosin filament is made up of 4 segments, so 4 protein chains, polypeptide chains. 2 polypeptide chains are going to make the heads, so one chain here, one chain there, okay? So those chains are called the light chains, okay? Each of the light chain is going to have 2 very important attachment sites. One of them is going to be attachment for actin. The other one is going to be attachment for ATP, okay? So one is going to be for actin, one is going to be for ATP. And again, you have 2 heads, so you have one of those attachments for each head. So that's your light chain. Now, when you look at the heavy chain, you're going to have 2 heavy chains, and they are going to coil around each other, okay? And this is going to make the tail of your myosin, okay? So here you have the head, and here you have the tail, and between the head and the tail, you're actually going to have the neck, okay? So head, neck, and tail. The neck is very important for the bending motion of the head of the myosin when you are undergoing a contraction and ATP usage, okay? Right. So the heads Heads of myosin are the parts that are going to cross bridge with actin and allow for contraction. Okay. All right, so that was your thick filament. So now let's look here at the thin filament. Is made up of actin, but is not a monomer of actin. You're talking about filaments of actin because they're like in a little chain connected to each other. When actin is connected to each other, you call that filamentous actin or F-actin. Okay, so it's right here. F-actin when it's in a little chain, right? If you are looking at only one protein or one globule here of actin, if you have only actin here that is like that, okay, so you call that globular actin if it is by itself. So one subunit of the filament is called G-actin for globular actin. Okay. Now, what's the story here with this actin? You do not see this actin alone in a muscle fiber. You're going to see that you have regulatory proteins that are associating with that actin. So the first long one that you find over here, this guy here is tropomyosin. The job of tropomyosin is to block the interaction of the actin with the myosin. Okay? So tropomyosin blocks the myosin. Okay? When tropomyosin blocks this active binding site here of actin, then nothing is going to happen. Okay? Now, what is holding tropomyosin in place is this other regulatory protein that is called troponin. Okay? So troponin and tropomyosin, they're both regulatory proteins. All right? So tropomyosin is blocking the interaction. Now troponin is going to regulate whether tropomyosin stays there or tropomyosin frees up the actin to interact with the myosin. Okay, so troponin, what is the job of troponin? It has three parts to it. Okay, so it is a blob that has three areas, three different parts to this protein. One blob, or actually a globular protein, right? One blob is going to bind to actin, okay? So that's going to anchor tropomyosin to actin. Another section is going to bind to tropomyosin, of course. So if it's holding tropomyosin on actin, so one part has to bind to tropomyosin. Another part is a sensor part that is going to bind to calcium ions. So when calcium floods the cytoplasm, it is going to go and bind to this subunit in troponin. And when you have calcium that binds here, this is going to cause a shift of the conformation of your troponin and is going to cause shift in the position of tropomyosin. So when calcium binds, then tropomyosin is going to come out of blocking your actin, and then actin will be able to bind to myosin. All right, so this, I know, is a lot of information in this slide, but everything that I mentioned here is important. Okay, so you really got to be comfortable and familiar with this filamentous actin right there. Okay, you got to be familiar, you got to be very comfortable with this orange molecule that is binding the actin, so that's tropomyosin. And you got to know also the function of these three blobs that is really composing the troponin. Right? You got to know what they're doing. Okay. All right. So here's another picture showing you what is going on, right? So filamentous actin. And this filamentous actin is being blocked by this stretchy, by this long fiber that is your tropomyosin. Okay. If it is blocked, it cannot interact with myosin, right? So here you're going to have your troponin, and troponin, you don't have calcium there, so troponin is allowing tropomyosin to block that interaction with myosin, right there. Okay. So remember, in your myosin head, you're going to have that actin binding site. But then if you have tropomyosin over there, then this site cannot interact with actin. All right. So here, looking at your sarcomere, okay, again, you have here your Z discs. You have this coiling thing. What is the name of that coiling protein over there? Titin. Okay, so titin over here, and over here connecting your actin filaments within filaments with this structure that is going to connect with the endomysium. That is not shown here, but that's going to be your dystrophin. Is going to be right there. Okay, I put dystrophin here. Okay, it's going to stabilize. Okay, your extracellular matrix with the sarcolemma. That is your membrane, and then it's going to be connected with your endomysium. All right. So here, specialized organelles that you're going to find. So one thing here that you're going to see is this sarcoplasmic reticulum. Okay, and your sarcoplasmic reticulum here is a modification of your smooth ER. Okay, so here is where you're going to have storage of calcium. Now, why is it you're going to have storage of calcium in there? Well, basically because inside of the sarcoplasmic reticulum, you're going to have this protein that likes to bind to calcium. It's actually going to sequester calcium inside of the sarcoplasmic reticulum. Okay, so unless you have a stimulus, calcium is going to be sequestered there and it's not going to come out. And what is the name of the protein that is sequestering the calcium? It's right there, calsequestrin. At least one name is easy to recognize, right? Job of calsequestrin is to sequester calcium where? Well, inside the sarcoplasmic reticulum right there. Okay. Besides your sarcoplasmic reticulum, you're going to find this thing that is a tube that goes all around here, your sarcomere, okay? It forms this disc in the middle of the sarcomere. So this tube here, you call it a T-tubule, okay? Now this T-tubule is actually part of the cell membrane. So it's a dip of the cell membrane that goes inside of the cell and interacts with the sarcoplasmic reticulum. So when you look at this dipping thing and this disc over here, it kind of looks like one of those hot water bags, you know, the old style hot water bags that you can get at CVS, like you put hot water in, you put the cap, and then if something is hurting, like your back is hurting or something, you put a hot water thing, right? It looks like, kind of like that, okay? So what happens here, you have an extension of your cell membrane all the way down here, and is connected, is sandwiched by sarcoplasmic reticulum. So you have here one slice of bread, the sarcoplasmic bread in one side, and then you have the filling of this membrane, and another sarcoplasmic bag on the bread on the other side. This formation here of this sandwich here, this is what you call this triad. Okay, so this is your triad. Why is this important? Well, basically, when you send a signal from your brain through the nerve hitting your muscle, that signal is going to be an electrical signal. It's going to be an electrical impulse. So you're going to have this membrane that is sensitive to electrical change. You're going to destabilize the voltage in that membrane. When you destabilize the voltage, you want to send a signal to your sarcoplasmic reticulum that this destabilization is happening, okay? The way that you can reach a lot of this sarcoplasmic reticulum at once is by having this type of formation of your cell membrane, okay? So this is going to be important for transmission of the electrical signal to a larger area inside of your muscle fiber. Okay, so electrical nerve transmission. That's the important function of the triad. Now the T-tubule. All right. So now looking at muscle contraction. So what's going to happen here? You're going to have sliding of those thick and thin filaments, and basically you're going to have this filament going that way, this filament interacting going this way. So the size of your sarcomere is going to change lengths. Okay, so the fibers themselves, the myofibrils, they're not going to contract in length. Okay, they're just going to slide on each other, and that sliding is going to allow this. Okay, elbows are Z lines. Okay, Z discs. So this is contracting. So you're going to be expanding, sliding, or stretching. Okay, this is the main idea of your sliding filament model. So what's going to happen here? This area that is called age light area is going to disappear. Okay, because your thin filament is going to reach all the way close to this middle section. Okay, so this area is going to disappear. Your A zone, that is a fat zone that is indicative of your thick filament, this is not going to change. Oh, let me go to another slide here. There you go. That's a lot easier to explain what's going on. Okay, so what is happening here is that you are going to have this I zone that is going to get smaller, is going to stay the same. The A zone is going to change shape. I always get confused with this part here. So let's see here in this picture what's going on. Now that's better for comparison. Okay, what do you see changing over here? Let's see here. What happened to your age zone here? The age zone is right in the middle of the fat A zone. That age zone disappeared because now you're going to have thin filaments that are going to be in the middle here. So this light area here is going to disappear. Okay, age zone is disappearing. Your I zone that goes here showing your thin filaments, that is going to get smaller because your thick filaments are going to invade the area over there, doing the sliding part. Okay, so two things are happening. The most important one really is shrinking of that thin zone. Okay, I zone is for the thin zone. A zone stays the same. A zone does not move. But this I zone is going to shrink. I find it confusing with this age zone right there in the middle. So I tend not to ask questions about that. Okay, if you really want to remember about that, be my guest. Important here is your I zone gets thinner during contraction. This is why I have here in red and bold and emphasized. Okay. All right. So here another diagram showing what's happening with the bands. Let's move forward. Okay. All right. So now we get to the nitty-gritty of what is happening biochemically with these contractions. Right. So you're gonna have to know everything about those action potentials. And you're going to have to know about changing this resting membrane potential. And you're going to have to know what causes that. Involuntary muscle contraction is a neurotransmitter that is called acetylcholine, right? So now we're going to get to the good part of this stuff. All right. So we start here with ion channels, okay? So what is happening here? Acetylcholine is going to bind to an ion channel. Okay, and this ion channel has binding sites, of course, for acetylcholine. If acetylcholine is not there, the channel is going to be closed. If acetylcholine gets there, then the channel is going to be open. What do you think is going to go through this channel? Ions? That's a good guess, right? Sodium. Sodium is going to be sodium. Calcium is not there yet. Yeah. Yeah, when acetylcholine binds, the channel opens. If acetylcholine is not there, the channel is closed. So this channel is very important for receiving the signal from the nerve and causing that sensing part, the responsiveness part of your muscle fiber. You're going to cause depolarization of your muscle fiber. Okay? The one way you're going to do that is by allowing acetylcholine that is coming from your nerve to bind to this channel that was closed. When acetylcholine is there, channel will open. So when the channel open, you're going to have an influx of sodium from the outside of the cell to the inside of the cell. So that is going to cause depolarization of your membrane. Okay? That depolarization is going to be transmitted from this channel that was chemically sensitive to another channel that is close by, but doesn't need the neurotransmitter. It doesn't need the chemical. It is going to feel the change of the membrane. It's going to say, this thing here, when it's negative, when it's resting, this channel is closed. When this potential becomes more positive, this channel will feel it and it will open. Okay? It's also a sodium channel. This thing here doesn't depend on the chemical, but it depends on the voltage. So you call this thing a voltage-gated channel, right? So you have two important channels here. You have a ligand-gated channel. You can also call it chemically gated channel. And you're going to have here a voltage-gated channel. When you look at them, let's say if you don't have these big green molecules of acetylcholine there, when you look at them, they kind of look like channels, donuts that have a hole in the middle and stuff will go through. So you don't really know how to differentiate them except for one thing. This voltage-gated channel has a little ball stuck to a chain at the cytoplasmic side. Okay? So this little ball stuck to the chain is going to be hanging around like this when nothing is happening and the channel is closed. As soon as the channel opens and an influx of sodium comes in, then this ball is going to say, oh, it's too much sodium coming in, so it's going to get stuck over here and plug that hole and stop the channel action. Okay? Important, very important function of your voltage-gated channel, sodium channel. Okay? Yes, I know, a lot of information, but hanging there, we're going to get to the whole picture over here. All right. So, what is happening here? You are going to have your nerve, voluntary nerve, that is going to come down and is going to innervate. So the nerve is going to divide here after the axon, is going to split and is going to innervate each muscle cell. So in order to excite each voluntary muscle cell, you're going to need to have this end of your motor neuron over there. Okay? If you don't have that, then your voluntary muscle cell is not going to contract. That's going to be different when you look at cardiac muscle. It's different. Cardiac muscle is its own thing. But these guys, the somatic or the thing that you can control at will, that is going to be receiving a nerve that has to get it excited. Okay? So this part of the neuron that is coming down and reaching the muscle, that is the axon. Okay. And when you get together of this axon with the muscle fiber, you call this a neuromuscular junction. Now there are two names for that. You can call it a neuromuscular junction, or you can call it motor end plate. It's the same thing. Okay. Oh, another question. In order to receive a signal to contract, each skeletal muscle must be served by what? Okay, served by what? Didn't even contract yet. He's just thinking about contracting. Okay, does anybody need more time? Everybody got the points? Okay, I'm gonna stop it. Okay, what is your answer? B, that is your nerve. That is B. Okay, so now we're gonna get into more details. Yeah, we gotta know this, okay? So here comes your axon that is coming from your neuron. And at the axon terminal, getting very, very close to your muscle, you're going to have a lot of stuff going on here. So the first thing that you're going to see is that you have these little vesicles. They're tiny, but they are not made at this place. They are made all the way up at the body of this neuron that sometimes is like more than a meter away from the site. Okay, so all the way up at the body of the neuron, you have these vesicle formations. Where do you get these vesicles from? They're budding out of something. Do you remember, like from cell bio class or biology class? Vesicles, they're transporting stuff. Go for a guess. Goji, yes, yes. You got a chocolate today? Are you sure? Oh, but I wanted to exercise, see if you get it. Okay, ready? Wah! Yeah, almost there. Okay, so Goji, right? You have your trans Golgi, you get little vesicles, and then you're gonna put them all the way down, all the way to the end of that axon. And how is it gonna get there? Transport molecules. You have those little guys. Have you seen those movies of those little guys? They get these big vesicles, comparison to the proteins, and they go walking like they have the tails, just almost like myosin, and they go like this, one foot at a time. The tails are kind of twisted like this, and they go like this. It must be torturous. So they get transported all the way down, right? When they get down there, look at this. They don't really have acetylcholine inside of them. They get packed right here, close to the neuromuscular junction. They get filled up with acetylcholine right here. We're going to see how. Okay? When they get filled up with acetylcholine, they are going to be ready to fuse with the cell membrane when the stimulus get there. So let's say that you are thinking about contracting the muscle, you send the stimulus, you send the electrical signal. Electrical signal comes down here through the axon and is going to hit another type of voltage gated channel over here. This voltage gated channel is going to allow calcium to come inside of this axon ending. Calcium is going to bind to a protein that is at the vesicle. The vesicle then is going to bind to the cell membrane and is going to dump acetylcholine right there in the middle between the nerve and the muscle. Okay, so a lot of stuff is going on here. Vesicles are going to fuse, it's going to dump acetylcholine right here. Now acetylcholine can go and bind to the chemically gated ion channel that is on the muscle side. Okay, you look at this muscle cell membrane, how it is all convoluted right there. It's full of wrinkles and dents. Okay, and there is a reason for that. The reason is that all around this surface area that is extended, you have a lot of those chemically gated channels. Okay, so when you dump your acetylcholine right here, you have a lot of channels that can pick up that acetylcholine. Okay, so this is the first part of sending the signal and fusing these vesicles and stimulating the channel that is across from your nerve ending. Okay, So that's your neuromuscular junction. All right. So the big picture here, what is going to happen here at the neuromuscular junction? You are going to excite those little channels, and you're going to cause something to happen inside of your muscle cell. You're going to send the transmission through the T-tubule, and you're going to cause release of calcium from the sarcoplasmic reticulum, and then you're going to cause your start of your contraction. So you're going from the nerve excitement to muscle contraction, and that is when you're going to get the cross bridge formation of actin and myosin, okay, and usage of ATP. So this is really the big picture that is happening for you to start getting your muscle contraction. And then, of course, there is more to this story. Okay, so first thing that's going to happen here, you're going to have at your axon ending, you're going to have those voltage gated calcium channels that are going to open right at the nerve. Calcium is going to get in, going to get fusion of your vesicle that has acetylcholine, okay, and release acetylcholine in your neuromuscular junction right there. So right there at the cleft. This is like a jelly-like substance, more like easy water, okay? So a lot of different charge, a lot of separation charge is happening over here that allows that to happen. All right. So acetylcholine diffuses, and then it's going to bind to receptors here at your sarcolemma, right? Membrane of your muscle, at the sarcolemma. And these acetylcholine binding channels, they are also called nicotinic receptors. Okay, they're called nicotinic receptors because they also respond to nicotine. Where do you find nicotine? In cigarettes. Yeah. Now, interestingly, you also have nicotinic receptors in smooth muscles, as we're going to see later. Okay. If you know people who smoke on a regular basis, you're going to find that they have this like morning routine, right? They get a cup of coffee, they smoke a little cigarette, and then 15 minutes later, they got to run to the bathroom. That's what happens usually. I mean, caffeine is going to stimulate some of the smooth muscle, but then nicotine is going to hit those smooth muscles responsible for peristalsis, the mean intestinal movement, and yeah, they're going to go to the bathroom. They depend, kind of like they get this dependence or reliance on this nicotine, right? So nicotinic receptors, cholinergic for acetylcholine, right? Nicotinic cholinergic receptors right there. All right. So remember, those guys here, those receptors are sodium channels. Close without acetylcholine. Put acetylcholine, they open up. Sodium is going to flood the muscle cell. Okay? When sodium floods the muscle cell, you are going to change the membrane potential at your muscle cell. Now, if acetylcholine keeps on binding to this receptor, you're going to keep on having muscle contraction. And that is not good. You want to have temporary impulse, and you don't want to keep on sending a message for your muscle to contract. I mean, imagine what was going to happen. Like if you want to, I don't know, punch a pillow. Okay, you're frustrated. You go study. I don't understand here. You punch the pillow, right? So you punch the pillow. You send a signal and you stop, and then you go with the other hand and you stop. Now, if you don't have the dissociation or breakage of that acetylcholine, then those muscles are going to be locked in a contractile position, and you're not going to be able to relax and then punch again. You're going to be like stuck in there, and it's like, I can't stop contracting this muscle. It's going to be painful. Okay? So this interaction here of acetylcholine with your cholinergic receptor here is temporary, is very fast, because there is a very important enzyme at your neuromuscular junction that is going to go there and break down acetylcholine. It's going to degrade it. Okay? What is the name of that enzyme? Something is an enzyme, you put ase at the end. Cholinesterase. Okay, that's the name of the enzyme. Cholinesterase is going to break your acetylcholine. And that's a good thing, because next time that you want to contract the muscle, then this channel is going to be open and you can send an impulse down again. Okay. Now, are there ways that you can stop the action of the enzyme? Do you know of an enzyme inhibitor, of a cholinesterase inhibitor? Have you heard of organophosphates? Pesticides. They block cholinesterase action. And then what happens to the pests? They die, because muscle contracts and they cannot relax the muscle, and if that happens to be a breathing muscle, you stop breathing. Does that happen to humans? Yes. Cholinesterase inhibitor is a toxin, or is a poison. I forgot the definition of toxin and poison. You can get killed depending on the exposure to these cholinesterase inhibitors. Okay? Just as a side kick. All right, so what's happening over here? First thing that's going to happen, your action potential is traveling down your nerve, and it's going to go to the axon terminal, and it's going to get all the way here to the neuromuscular junction. Close here to the neuromuscular junction, your voltage-gated calcium channels are going to open at the neuron side, okay? Then you're going to have fusion of your vesicle with the membrane. You're going to get release of acetylcholine. Then acetylcholine is going to bind to the receptors. Once you bind to those chemically gated receptors, you're going to open the sodium channels. They're going to flood your inside of your muscle cell, and it's going to change the membrane potential over there. Okay, it's going to activate, it's going to allow excitement of your muscle cell. Then you don't want to keep that excitement forever, so cholinesterase, okay, acetylcholinesterase is going to break down your acetylcholine at the cleft in this jello soup right there. Okay, here in the cytoplasm, okay, you're going to have acetyl-CoA. Where you get acetyl-CoA from? Glycolysis, right? Going into citric acid cycle, right? So you get acetyl-CoA there, high energy stuff, right? You get acetyl-CoA, and then you get a molecule of choline, okay? And you have this enzyme that is going to paste those two things together. You call that choline acetyltransferase. You get acetylcholine right here in this cytoplasm, okay? But that is useless because you need it inside of the vesicle, right? So acetylcholine is made in the cytoplasm, and now this vesicle is very much charged with hydrogen, and you need to get acetylcholine in there. You don't need hydrogen there, right? So what you do, you have this other channel that is called an antiport channel. Remember, antiport channel, it exchanges things from one side to the other. So what this antiport channel is going to do is going to use the proton that has high gradient, high concentration inside of the vesicle, and is going to exchange. It's going to kick out one proton, is going to allow in one acetylcholine. Okay, so this antiport movement is going to allow packing of acetylcholine in high concentration inside of this vesicle. Right? And this antiport channel is very important because if you would just open a channel for acetylcholine, you wouldn't be able to concentrate it against its concentration gradient. Okay? So this indirect form of energy usage is important for packing acetylcholine inside of the channel. Right? So keep in mind what happened here. First thing, you had electricity that came here, was transmitted down the line. Calcium channels that are voltage gated opened up. You have these already packed in there. Now calcium is going to bind to molecules that are in the vesicle and is going to allow membrane fusion. Right? What are the names of the proteins? They're T snare and V snare. Those two molecules at the vesicle and at the membrane, they are going to allow membrane fusion and dumping of that chemical in the neuromuscular junction. Okay. All right, I know I'm running out of time here. So make sure you understand everything that is going on until here. Because when these sodium channels open, this is when you're going to start this propagation of this signal to your muscle. We'll pick up this on Wednesday.